Good. Yeah, sorry for the delay. I had some issues with traffic. That's why I was running. So before we start, did you guys start reading the paper or not yet? Do you have any questions? Not really. Okay. So today's lecture should be somewhat short, depending on the discussions we'll have. The reason why it's short is because the course material is divided into topics, and so this one is a little bit short. So we might finish a little bit earlier. I'll also be presenting the team project that you'll be doing. So I guess this is the time to choose your teams and, get started on it, or at least finding the right paper, because you'll be trying to agree on a paper that you'll present. You'll send it to me so I can approve it. And so that takes a little bit of time. I'll put a deadline on that at some point, but it's a good opportunity to maybe choose your teammates either today or whenever you want to. I think we're old enough to do that. You don't need me to guide that stuff. How many people is there? So 2, 4, 6, 8, 10, 12, 14, 15. So we're missing 3 students. So I guess I can put those students together, and that's pretty much it. So it will be teams of 3, okay? So you can do that today. What else? Today we'll have also the homework that's going to be assigned. So the 3rd homework, I believe. You'll get your grades for the other homework hopefully by Friday, so that you have enough time to adjust the next homework, okay? And then after that, there's only one left. Following that, it's only the 2 articles, the 2 essays, plus the presentation, and obviously midterm and final, okay? Questions for me? Easy. Okay, what else did I want to say? Yeah, I think that's pretty much it. Okay, so today we're going to continue on the topic of getting the human genome. So last lecture, if you remember, we were looking at characterizing the human genome by doing some karyotyping, and that's important for multiple reasons, which I'll review in a second. Today we're going to be taking a look at how we can construct a library from that human genome. Obviously, this is a bit different from, well, the rationale behind it is the same as yeast. It's just now we have a degree of complexity that is much higher, and so we need to have some considerations in place before getting the genomic library. So we need to kind of optimize the way that we were working so that we have a much more simpler approach, because when you increase the amount of nucleotides to be sequenced, and then obviously the human genome has a lot of repeats, you end up having a little bit more complexity, and so you have to update the method that was used for yeast. Okay. Next lecture we'll be looking at mapping and contig assembly, which this year is not found for, which was not used in the Craig Venters approach. But since we're looking at the Human Genome Project, we'll be taking a look at this. And then finally, the sequencing. So we'll take a look at some techniques. Obviously, annotation, I'm not covering this part too much in depth because Amandeep is doing this in 512, okay? But I'll still talk about it fairly quickly just to give you an idea of what it is. So I was watching the lecture. By the way, is there anybody watching the lectures online or kind of? Okay, good. So the lecture, I was rewatching it just to make sure I'm not saying random stuff. And I think one of the things that was not highlighted properly last lecture was, what's the point of karyotyping really? Why aren't we just directly breaking up the genome and doing, going through the contig assembly and then sequencing? So I'll give you a few examples here of why the karyotyping is important. So first of all, just a quick review of karyotyping. We talked about 2 methods, the GIMSA and Fisher method, which Fisher is a bit better because it gives you these colors here, which allow you to kind of see translocations much better, but not inversions, which are within the same chromosome. And GIMSA, it's pretty much the opposite. And so Imagine you have a genome that you're trying to sequence de novo. So that means you don't know anything about it. So the idea about karyotyping is that you will figure out its chromosomal setup so that then you can make sure that you are sequencing the right thing. Why is it important? Because if you take a cell and you break it and you don't know its karyotype, so the content in terms of nuclear chromosomes, then for example, if you have a duplication in your genome, let's say for example a region of a chromosome is duplicated twice in the genome from evolution or whatever, you will sequence it and it's the same sequence. So you don't know if that sequence is only on one chromosome or two. Okay, so this is why you need to know the content in terms of chromosomes and then their patterns as we've seen in the GIMSA, because that will allow you to see if your sequence is only like one single sequence in the genome, or is it a duplication that exists on two separate chromosomes. That's a good example. And so if you're sequencing this part that is similar or identical, then you don't know if it's occurring once, twice, or multiple times. And there are many examples of this in the genome. Actually, the human genome has a lot of repeats in it. And so those repeats, you cannot really, the sequence remains the same. So you don't know if it's on one chromosome or 10 chromosomes have the same repeat, etc. So this is one thing. Obviously, we've seen translocations, deletions. We've seen translocations and inversions. We didn't see deletions. And I'll explain a little bit about this, why we need karyotyping for pre-sequencing. When you're taking a genome, so when you're trying to sequence a human genome, you can't just take the human genome from one person and go and sequence it. Because that person might have a specific pattern of DNA, but that doesn't mean that, let's say, for example, the genome from me will have the exact same pattern. We have mutations that happen, we have deletions, insertions, translocations. And even though they're not that big, unless you have a kind of some sort of genetic disease or whatever caused by these, but our genomes are not identical. And the Human Genome Project, for example, they took DNA from multiple different ethnicities, multiple groups around the world, and sequenced them. So then if you're trying to sequence this, and you have deletions in one group, particular group, but not the other, how will you know where that part, where that sequence goes? So you need to karyotype it to know that in that chromosome, for me, it's not there, but for you, it's there, right? And then you have translocations where, for example, me, I have a sequence of DNA that is on a certain chromosome, but because that sample that I took, that person has some sort of disease where a translocation happened, then you need this karyotyping to be able to say that this part for this person went there. Okay, so this is why karyotyping is quite important. Actually, it's you need it. You can't do without it. Okay. It also confirms ploidy and chromosomal composition. So this chromosomal composition, I just said it. Ploidy. If you're trying to sequence a human genome, we are diploids, and we have the sex-linked chromosomes. So you need to know what type of chromosomes are there. In terms of ploidy, you don't take the same approach for sequencing if you're trying to sequence a tree, which could be triploid, tetraploid, to a human, which is diploid, to a yeast that has two phases, haploid, diploid, right? The reason for that is that, for example, here, if you take a look at chromosome 9, well, you have double the DNA sequence. And so it's important to know if your sequence is occurring twice, 3 times, 4 times, whatever. And obviously, if it's an X chromosome or a Y chromosome, then that adds a level of complexity. So you need to understand this composition. So this is why karyotyping is important. The other thing is, your goal is to assemble the genome. And part of assembling the genome is knowing what are the major parts of it, right? You need to know which chromosomes are there, and then how are they segmented while you're doing your stains, for example. Just by the stains, you can know that, for example, a certain part is the first one, and then the second one has this specific pattern, and this one the third pattern. And so you can create these like primitive maps, let's say, of chromosomes. Okay, and this is important because our goal is to assemble the genome sequence. And so we go with a low resolution method, which is the karyotyping, towards a high-resolution method, which is the actual sequencing itself. Okay? Simple? Easy? Okay, so let's get back to the genome sequencing that was done by the Human Genome Project. I was going to present the paper. I forgot how big it is, and then I gave up on that idea. The paper, I think, is like 100 pages long or something like this. So if you want to read that and see how the hell they did it, you can do that. I got to the first figure, which I'm using here. The others, it's a bit more in depth, and we don't need to go into that. But the genome sequencing approach for the Human Genome Project went as follows. It's called hierarchical shotgun sequencing. Shotgun. So if you're doing any technique, any research approach that says shotgun, shotguns means just throw everything together and try to figure out what is in there. For example, shotgun lipidomics means you just throw all the lipids in the mass spec, and the mass spec will give you all the peaks, and then you try to figure out which peak is corresponds to which lipid, for example. So in this case, shotgun means we're just throwing everything together and then we're sequencing that at the same time. Hierarchical, it's because we're breaking the DNA molecule into smaller pieces, and they are hierarchical because you have a big piece like this that you will cut in 3 parts or 4 parts, and those are contiguous sequences. So this is the hierarchy, from a large piece to fragments that you put in plasmids or whatever, and then to sequenceable, I don't even know if that's a word, but sequenceable fragments, okay? So this is the hierarchy. In this specific project, what they did is that they took the genomic DNA, they broke it into what's called a BAC library. BAC refers to bacterial artificial chromosome. So this is where you're actually inserting this DNA into an artificial chromosome. We'll be taking a look at this a bit later, maybe slide 20 or something like this. And then what you're going to do is create your contigs and then break those contigs and then shotgun them, sequence them, okay? And it will give you DNA that is overlapping like this. This is your contiguous sequence, which you will actually just use a computer to kind of remove from here and then assemble everything, okay? So the process is quite simple as shown here, but actually it's just a lot of work. That's pretty much it. That's a lot of work, and we'll be taking a look at all this stuff here. So first, as I said, you start with a double-stranded DNA. You will break it into pieces. Now, when you're breaking it into pieces here, this diagram says restriction nuclease, but actually there are 2 methods that you can use, either a physical method, like ultrasonication, etc., or an enzymatic method with these nucleases. Then you have these fragments. These fragments, you have to insert them into some sort of recombinant plasmid. And now there are some considerations to take in here at this step. There are some other considerations to take at this step because we have sequenced the influenza bacterium. We have sequenced the yeast, Saccharomyces cerevisiae, but those had only a few million base pairs. Bacteria has 4.6, I think, and then the yeast has 12. We have 3 billion base pairs. So that's 100 times, a bit more than 100 times bigger. You don't necessarily want to make 10 million or 100 million plasmids, right? It's almost impossible. Okay, so you need to review your methods and to optimize them so that you can create your genomic library. For the vector that you're going to use, you can't just use bacterial plasmids. They can only take up about 10 kilobases. If you divide 3 billion by 10 kilobases, you'll realize that you'll have so much plasmids, and then of those plasmids, which ones are good, which ones are not. And so you have to switch it up. There are multiple ways you can use bacterial plasmids if you have a lot of time and thousands of people. You can use phage transduction, which we'll see right now. Shuttle vectors, cosmids, phosmids, yeast artificial chromosomes, which are inserted into yeast and not bacteria. So again, this is just a correction to introduce plasmid into bacteria. This step here is for yeast. And then bacterial artificial chromosomes, and you'll see that basically the best one is the BACs of these. Okay, so we'll go in order of which one is the best until we reach here. Again, with me creating problems and then solving them at the end of the, the lecture, okay? Before we start, participation. Let's do a little bit of, let me test your science. Okay, so when you're trying to build a genomic library, you can't just jump in and be like, okay, I'll break it, I'll do this, and put them together, sequence, that's it. You have to think about the whole project, and this is just the thinking that you do before every experiment that you will be doing in your whole life. So you need to think about each step specifically. One question that you have to think about is, your goal is to create a library that represents your whole genome. So it has to be comprehensive. For that, you need to cut it. And if you cut it too much, or not enough, you'll have stretches of DNA that are not of the right size, and that will lead you to missing the fragments. For example, if you're not cutting enough, and you end up with fragments that are 100 kilobases, then you try to put it into a simple, small vector, it will not take it up. And so you need to think about how do you solve that. So, do you have any answer for me for this one here? Yes?
We can try to, let's say, fragment it and leave it for different times, and then just do like a gel for each and see which vial creates the stretches with the optimal length.
Okay. This is for physical or restriction enzyme? Anybody? Yes.
What she said?
Oh, it's okay. You can.
Maybe physical. Like if we test if it's the right length, and if it's not, we put again in the physical shearing.
Okay. So, does anybody have anything else to add? Yes.
We can use multiple restriction enzymes, so we don't miss any fragments.
Okay. Julian?
I was going to say that, but also in addition, you can use a PCR to have a greater volume of samples of DNA and then reproduce the exercise multiple times with multiple different restriction enzymes and then compile the data to get the pieces together.
Yep. So let me first write your names so I don't forget, because I have a few questions here. Okay, so we have Alessandra, Arthur, Julian, and what's your name? I forgot. Sarah. Yeah, same first name and last name starts the same way, has the same letters. Okay, I will remember. Did anyone else answer? I think that's it. Okay, so let me just go over the answers here so that everyone understands. So we have two problems to solve. First, you might miss fragments. So to avoid missing fragments, what you can do is increase the amount of starting material, right? But that doesn't solve one of the issues, which is too big. The fragments, if they're too big, then you end up missing them anyways, even though you have, the plasmids can still take up a huge amount of DNA, but it's very rare. So if you increase the amount of it, what's going to happen is that you have a chance of getting that fragment, but you also can use multiple restriction enzyme at the same time. The problem with this is that it's very hard to predict the fragments that you will be producing, because if you cut with 2 or 3 enzymes at the same time, then you end up cutting fragments into smaller fragments into smaller fragments. So that's problematic too. Now, one way to do it is to actually optimize your digestion time and the amount of enzyme that you put in there, so that you can have multiple different time points. Let's say, for example, fragment for 1 hour, 10 hours, and 100 hours, and then see which ones give you the fragments of the right size. So that's the solution. If you have physical shearing, as we'll see in a few minutes, the physical shearing method will give you fragments of approximately the same size. So then that's just a way to get them all the same size, but then you'll have to modify those fragments because there's no sticky ends at the end. So you can't ligate them onto chromosomes very efficiently. You can still do it, but not as efficiently, so you need to modify those. And we'll take a look at that in a bit. Question number 2 that you could be asking yourself before building the genomic library. So a plasmid is not your best buddy. He's not just going to take the fragment that you give it and that's it. Most of the time, the energy efficient reaction will be just the plasmid opening up and then closing back on itself. How do you solve that problem to ensure that it has an insert? Yes, Sarah.
For example, using the different antibiotic resistance and use the blue and white screening, that's what we learned. So there's going to be this marker there, kind of.
Yeah.
Kind of different markers. Can you just check for it if it's there, then we look up if it's not.
What if none of the fragments get inside the plasmid, and the plasmid is so fast at closing up that all your bacteria die? Wait. Yes, Alex, right?
I think it's called RSAP you can use. It is a phosphatase or kinase that alters the ends of the insert region of the plasmids that doesn't do that.
Yeah.
Additionally, you can just check on a gel after you're done to see you cut it with an enzyme and check it's the length of the insert, or sorry, the length of the vector, and then just toss that clone.
Yeah, that's a very good point. I have a slide on that, so you just killed the slide. Yes, Sarah.
We can use cellular sequencing to confirm that the insert is cloned into the plasmid. It's only a confirmation.
So you're sequencing the sequence. That's a good idea. So when you have a plasmid, yeah. So I will explain what she said. So basically, you have the plasmid. You know the sequence of that plasmid, so you can create primers before in the sequence that you know, and that will tell you if there's new sequence that appeared in the middle or not. Okay. Very expensive way of doing it, but actually the way that tells you exactly how it inserted. Because what happens is that if you have a plasmid and you try to insert DNA into it, sometimes the DNA will flip and go the other way, right? So this is not really a problem for sequencing because the DNA can be read from here or from here. But if you're doing research and you're trying to study the expression of a certain gene or protein that you are interested in, then the direction matters because your origin of replication or your promoter will start in this direction. And so if you flip your insert, then it starts with a stop codon and that's it. Okay? So basically that's that. Let me just write the names. So Alex, Sarah times 2, and then Sarah. Who else? Yes.
For the flipping, there's actually like restriction enzymes you can put like in this way so that it can't
Flip.
flip.
Yeah. So you can cut with different enzymes to avoid the flipping, because if you have an enzyme, so there's a way to make sure that that fragment has only one direction of inserting, and that just depends on your choice of restriction enzyme. Yes, Arthur.
It's a question. If we get two plasmid with for example, two same restriction enzyme, can the both plasmid stick
Fuse?
together? Yeah.
Yes, they can fuse together. So you can join plasmids together if you want. I've never heard someone doing this for fun, but probably they can join. Yeah, if you use the right ligase and the right sticky ends, you can stick it pretty much anything together.
Yeah, I was more saying in a way like be careful that the plasmid are not joining together.
Yeah. So. Okay, I see what you mean. So, because you can have two empty plasmids that just join together, right? Yes, it is possible, but again, there are reactions that are more favorable than others. The religation with itself is the most favorable because in terms of stoichiometry and the position of the nucleotides, it's the easiest one to do. The insertion is the second, and then two plasmids fusing, it's like the least, but it could still happen. Okay, who did I forget? Yes, you. Maria, right? Okay, cool. Then after new teacher's way of doing it on the phone directly. Okay, more problems. Another thing that you have to think about, as I told you that it could be a problem, is the fact that your size of your fragment could be too big. And if it's too big, then you end up having the smaller fragments dominating. This is just because the ligase activity is more fit for smaller fragments. It's easier to handle. You have to think about the DNA as moving and then the ligase trying to do its job. So a smaller fragment creates less force and does not disrupt the ligase. Plasmid size constraints. So you have a small plasmid. They have limits in terms of how much DNA you can put in there. Again, just because of the physical properties of the plasmids. And this one is a very interesting one. It's the transformation efficiency. So your bacteria, you will make it porous to allow for the plasmids to go in. Now, obviously, something too big cannot go in, and so transformation efficiency is affected by that. So even though you're creating different plasmids of different sizes, if the insert is 10 kb compared to 2 kb, then that 2 kb is a small plasmid, and that will have an easier time to be transformed into the bacteria, while the 10 kb plasmid requires bigger pores or actually is not even taken up. Okay, so how do you solve this problem here? Yes, Gabrielle. Gabrielle?
Gabrielle.
Yeah.
You can use a different plasmid, like a different vector, like what was on the other slide. Like
Yeah. Any other way? Yes, Akil.
Using one way is using different strains of certain organisms have different transformation efficiencies. Some people engineer certain strains, like remove certain like restriction enzymes to increase transformation efficiency. So that's one way, and another way is to just optimize your protocol, whether it be electroporation or chemical transformation or whatever it might be, to I guess optimize the transformation efficiency with the bigger plasmid.
Yeah. Mark. No, Matthew. Jeez, Mark, Matthew. Yes, Matthew.
I think I remember reading that they used to plate all the transformants, and then there was a way you could like blot the culture plate and then run a PCR on that, so you could see which colonies had which size inserts in them, I guess, and then you could just select one of each colony, and that way you get an even.
Yeah. That doesn't solve the problem of bigger fragments not being taken up, right?
Just not being taken
Yeah.
at all.
So then for that, you would have to do, I'll repeat your answers. Mark, do you have anything to add? Okay. What did I want to say? So, man, I like this class. Give them some points for participation and they wake up. I'm used to teaching 200 level classes, so the students are very shy. They don't know yet, but you guys are giving me good answers. That's good. So, transformation efficiency. Let's start by this. So that's a very good point that Akil had, which is some strains are better at taking up DNA than others. Most of the bacterium that you'll work with in the lab is a strain called DH5 alpha. But there are different strains that have different transformation efficiency, and then depending on your plasmid, you'll try different strains. So this is a good point. Obviously, you can just optimize your reactions, as Gabrielle and Akil said, by making sure that you're using the right vector for the size of your fragments, or by also optimizing your shearing methods. Okay? Yes?
Why don't you just use another cloning method?
Like what?
Another method of cloning.
Like?
Gateway.
Gateway. Did we have Gateway at that time? We're in 2000 something. 2000, yeah. Obviously, Gateway is quite expensive. Do you have experience in lab? Okay, so that's why the Gateway. So Gateway is a very good way of cloning stuff now, but at the time it's not. It's not the ones that were mostly used. I think Gateway has been there for a while, but it's very expensive. So I guess that's one of the reasons. But probably there are other reasons that I'm not thinking about why Gateway was not used. Okay. For this specific project, the Human Genome Project, this is what they did. I'm presenting it basically now. Sarah taking all the points. Okay, so let's start answering a little bit those questions, and some of them that I did not mention. So Alex has mentioned this one here. So if you want to ensure that you have inserts and to avoid that your plasmid closes back on itself, what you can do is use some kits that will just do a dephosphorylation. So you have your DNA, your circular plasmid, you cut it, then you have phosphate groups at the end of both sides. So what you do is you dephosphorylate one of the sides and you allow for the other one to stay there. And what happens is that when you're about to do the insert, the other one that has the phosphate group will allow for the reaction to ligate both DNA strands, the plasmid and the insert. I'll repeat this, okay? Imagine the plasmid like this. You cut it open, okay? I see some faces like, what the hell is happening? So the plasmid is like this. At the end, you have your 5' and your 3' DNA side, okay, of your DNA molecule. They have a phosphate group, and it's that phosphate group that allows to add the next nucleotide and the next nucleotide. So to avoid the plasmid religating on itself, you will dephosphorylate one of the sides, and you leave the other one phosphorylated. And that basically prevents them from going back together. And so when you have it open like this, the side that is still phosphorylated will allow for the reaction to ligate between your insert and your plasmid. Make sense? Okay. There are kits that exist to do this right now. Before, you just use a phosphatase. Try to optimize the reaction time so that you don't dephosphorylate everything. And then obviously, there's because your strand has another phosphate at this side, then what happens is that you have your plasmid with the ligation here, and then because there's the other side that has the phosphate, it brings everything together, okay? I think Sarah mentioned this. The other way to do this is you can use a detection system in the plasmid. For example, I'm just showing you something that you learned, I think, at least 10 or 20 times. This is the PUC19 plasmid, which is not used that much anymore because it's very inefficient. But it has a LAC-Z gene. And this LAC-Z gene here, when you add XGAL to the plate, the colonies become blue, okay? If you put your insert, so if you cut here, for example, using, I don't know, bagel 2 here, and you put your insert in here, then you break the protein, the LAC-Z protein. And what happens here is that because you broke it, you do not make any beta-galactosidase enzyme, and so that will make the colony not react to your XGAL. And so the colony will be white. So this is one way of making sure that you have an insert, because only plasmids that have an insert will have white colonies, the others will all be blue, because this was not disrupted or it religated on itself or whatever, okay? What we do now, which is a little bit more simpler, because here you still end up with the same amount of colonies and you don't really want colonies that are useless. What you can do is use a toxic gene instead of the LAC-Z, right? One example is a toxin called CCDB, forgot what it is, but basically the toxin, so if you don't have the CCDA enzyme, you are sensitive to the CCDB toxin. And so what happens is that when you insert in the middle of the toxic gene, you're basically preventing it from killing the cell. So you use a strain that is sensitive to that toxin, right? And then those that don't have the insert will have the right sequence, so they will have the right toxin, and that toxin will kill the cells. This way you end up with a more filtered lawn of bacteria that has only your inserts in it, okay? What the hell? What did I write this here for? Insert into DNA. Oh, yes. Okay, so this we'll see in a second, but basically, for example, if you're taking a look at phages, the phages require a specific amount in their capsule, in the head. So if you don't have that specific amount, basically your insert plus whatever DNA that the phage has, then the capsules will not form, okay? And we'll see this in a second. I'll explain more about this in a second. Okay, so we're dealing with human DNA. So we have 3 billion bases. We need to find, we need to clone big fragments. And for that, we need to change the way that we do this. So before we used the standard cloning in plasmids like this guy here, only allowed you for around 10 kb plasmids, but this does not work for our situation right now. We need much bigger, much better methods at carrying the DNA that is much more longer. Okay, it's supposed to be about 300 kb to 1 million base pairs. So obviously you cannot fit that in here. Okay, so how do you do that? Now is the solution. So you change the vehicle that you're using to carry the DNA. So the first and the least efficient one of those methods is the lambda phage. The lambda phage has the head here, the tail, and usually it has these legs here or some sort of needle that inserts the DNA into the cell. The lambda phage puts about 50 kb DNA into the head, and this DNA has to have these COS sites, which are cohesive sites. So it's just sites that can ligate back together on both sides. So if they're there, they will build this circular plasmid, and then if you break them, they will make a chromosome. Okay, so phages have a lot of unnecessary DNA. Again, just reflecting on the other stuff that we learned, there's a lot of stuff that you can remove from organisms and still allow them to do their job pretty well. So in this case, what you do is that you remove the unnecessary lambda DNA, and this allowed you to insert foreign DNA, which is your fragment of interest, which is about 20 kb. So there's 20 kb of useless stuff in the lambda phage DNA. The other 30 kb, you need it to carry on the job of the phage. One part that was replaced is the stuffer fragment. And this stuffer fragment is just the protein coding fragment of the DNA that stuffed DNA inside the phage head. Okay, and so you can remove this and still optimize your reactions to still ensure that your DNA is inside the head, and if it's not, then you don't use it. Okay, so this is the part that was removed. It's called the stuffer fragment. Here's what it looks like. Let me just show you something actually cool. If you try to understand stuff about figures, I don't know if you guys are aware of this, but I'll show you something here that you can do and then start learning. So this is one thing that's useful, that you can actually use this tool for it. So you copy, okay, let me go slowly because I'm using short keys and all that, and then you paste it here. Wait, what the hell? This guy wants to cheer up, man. I think I have to use my account because, okay, let me just log in because I have to use my own account, okay? Jeez. You know what I'm tired of? These two-factor stuff. Yes, go ahead. And then 53. Does anyone know what I'm about to do? No? Okay. 50, yes, it's me. 50, let's do it, buddy. Okay, so if you're paying, you can copy and paste the picture in here, put it here, and then what's going to happen is that it will tell you exactly what's on the picture and actually explain every piece of this thing, okay? So if there's something that you don't understand or you're trying to get from a picture that has no explanation, you can use this here. And obviously, you can ask it to explain more stuff on this, okay? So this is a pretty cool trick that you can use for studying. Sometimes I use it whenever I'm trying to understand something from other people's slides, like this professor. So I just use this, and then it will explain to me whatever the hell is in here, okay? So back to this. So, 1 second. Jeez, I have to move this stuff here, go there. Don't ask me why I'm paying, okay? I use it a lot for coding. So this is the plasmid. This is the lambda phage genome. So you have the genes that are important to produce the head, the tail, and then in this section here, which is the recombination section, you can put your own fragment of interest. Obviously, you have different promoters, and you have DNA gyrase. I think it's this one here, if I'm not wrong. And this DNA gyrase, all it does or gyrase. All it does is that it turns the DNA into supercoils so that you can put them in the head. What's important for us is not all this, it's the recombination part, which is right here, and then you have also the cause sites, right? And so whenever you're trying to package the plasmid in the phage, it will be in a single-stranded, in a double-stranded DNA molecule like this. Whenever you put it in the bacterium, what's going to happen is that this section will stick with this section, and then you have your plasmid. Okay? So this is the lambda phage. Now, why am I presenting this guy? So what's cool about lambda phages and why they could be interesting to for genome sequencing and library generation, it's because it gives you these high efficiency libraries. So when you're using a phage, the phages don't form unless there is a specific amount of DNA in the capsule. If the DNA is not about 50 kb, the capsule will not form. And if the DNA has no insert, then the DNA will be only 30 kb, and that is not sufficient for the head to form. So what you end up with, only inserts inside the phage, okay? Mostly. Sometimes mistakes happen. The inserts are about 15 to 20 kb long, so this is not sufficient. But depending on your purpose, you can use these phages, and you can build them in vitro. That means you can do this all in a test tube without having to use a cell. Problems with it, so one of the problems is that you are doing this in a lytic phage, so you will be forming plaques like this. This you've probably seen it before. Just quickly, so these are all bacterial colonies that were lysed with this phage. So that means there were inserts inside the bacterium, and then the bacterium was exploded. And when that happens, you have your plasmids that are right there. Okay, so these are the plasmids. Well, this is basically the bacterial colony that is broken up, including these structures here, which went from here to here because of the cohesive sites, the cause sites. You can screen the clones of interest by using hybridization or PCR. Hybridization is where you have a certain probe that you will just put on a nitrocellulose membrane, and then your plaques, the ones that have the specific insert, will light up under, X-ray or under fluorescent, whatever method you're using, or PCR. But these require you to know a little bit about your DNA molecule before doing that. This is where Sarah's answer comes into play. You know what the plasmid looks like. You don't know what your insert is. So you could technically create primers or probes for this part here and this part here, and then use that to probe for this sequence here. Okay? So that's that. Not the best, but it's still used. It's still used in research. It's just not interesting for genome building. The next methods that exist are cosmids and phosmids. So these are different vectors. Cosmids is just a mixed words of plasmid and cohesive sites. So you take the plasmid that we learned previously and you add to it these cohesive sites at the end. And so this will allow it to be linear and circular, and then you can add DNA to it and then package it inside the lambda phage. Now, what's the difference between a cosmid and the lambda phage's own plasmid or genome? It's that the lambda phage has a lot of DNA that you technically don't really need. And so what you do is that you will take your plasmid, which has less useless DNA, because it basically just has this here. You add the cohesive sites here and here. And what happens is that now you can insert your DNA and you have a selectable marker with it, which you didn't have in the phage DNA. And you have a bacterial origin of replication in it also. Okay? This allowed for bigger inserts because the plasmid's own DNA was not that much, and then you have 40 kb that's left. Cool? Easy? I think we're going to do the full lecture. Phosmids. So phosmids is, I don't know if you guys remember from genetics or any other class. So bacteria, to transfer DNA between each other, they have this F plus plasmid, which is the fertility factor. and then that allowed you to transfer DNA from one F-positive bacterium to the other. It's like their sexual reproduction, kind of. And so the phosmids come from there. So you take that F plus plasmid, and then you add to it the DNA of interest. Now, why is this an upgrade? It's because the phosmids, usually you have one or two inside the bacterium. You don't have that much. And so it's a low copy number plasmid. And because it is a low copy number plasmid, it prevents you from having changes in your genome. And I'll explain what this really means. Human genome is highly repetitive. You have a lot of DNA that is repetitive, and that will lead to problems because there is rearrangements. Okay? And so when you have rearrangements happen only when you have DNA that is replicated. And so if you have a low copy number, you have less instances where you have making duplications of the same DNA material. And if you have less duplication of the same DNA material, you prevent the rearrangements to happen between two repeats of two different molecules, DNA molecules. Does that make sense? So for example, if you have a high copy number plasmid, which is the cosmid, for example, your plasmid will be replicated thousands of times. That increases the chance that two copies of the same fragment will have translocations between them or inversions or whatever. Okay? And the human DNA, as I told you, has a lot of repeats. And so you want to avoid that. This is an example here. This is a commercial kit where you can buy it and actually use it. And they are used in research right now. It's called copy control. This is the vector. And then basically it follows the exact same way. So what you do is that you'll break your DNA, ligate it using their vector, and then it will give you plasmids like this. And these plasmids are the ones that you'll use inside the phage to then destroy your bacteria and produce many copies of these. Okay, and you can create your library from this. But they're useless. They're useless for the genome sequencing because they still create between 40 to 50 kb long fragments, so that's not very interesting. You need bigger inserts. So one way is to do it through the yeast artificial chromosomes. So we had to create these chromosomes ourselves, or the bacterial artificial chromosomes. And so in the Human Genome Project, they used the bacterial artificial chromosomes, and we'll see why. The bacterial artificial chromosome allows you to put up to about millions of base pairs, but usually it's about 300 kb, okay, that you can guarantee, right? Start by the yeast artificial chromosome. So if you take a look at this picture here, you can try to understand it, or you can copy paste it and then get all the information from it. So now I'm going to explain it to you, but if you look at it like this, it's quite hard to understand what the hell is happening. And so you could use the trick I just showed you to do that. But yeast artificial chromosomes are special because they allow for the DNA fragment to be inserted in the form of a chromosome. And chromosomes, as you know, they can have a huge amount of bases, okay? So a few features of this chromosome before we start into how you produce your artificial chromosome. It is a plasmid that has the ARS, autonomously replicating sequence, and it has a centromere, so right here, and it has telomeres here. So the telomeres are basically the end of your artificial chromosome. So these sequences, you can take them from anywhere and add them to your plasmid. Centromere, it's important because when you put it into yeast, this plasmid, so this is the only plasmid that you put in yeast and not bacteria, okay, from everything that I showed you. The centromere allows this chromosome to be replicated as another chromosome, a naturally occurring chromosome. So this allows you to be a little bit more stable and have proper segregation of your inserts between the different yeast cells, okay? You have a selectable marker, the uracil 3, which allows you to grow on uracil lacking media, and then obviously some resistant genes and origin of replication for the bacterium, okay? So what you do, you take your DNA and you break it into your genomic DNA, you break it into multiple pieces, and you try to purify 400 to 500 kilobases. Now we're getting to the real amounts that we're interested in. And then you have to size fragment them using pulse-filled gel electrophoresis. I'll show you what this means in a second. You also digest your artificial chromosome plasmid using different enzymes, for example, BIMH1 and EcoR1. And this will allow you to open your artificial chromosome like this. And then what you do is that you stick them together, okay? And these ends obviously have to match the ends here. And that will allow for yeast to ligate this huge piece of DNA inside the artificial chromosome. And then the centromere will allow it to be pulled between cells. And the telomere is protected from degradation and all that. Make sense? Yep.
What's the relative size of the artificial chromosome?
Very good question. Let me see. So just by looking at the content here, I think it's like 5, 6, no, should be maybe max 10. You know what, let's ask our friend here. Maybe it could be useful. How big is the YAC chromosome without insert? Cool stuff. 15 kb. So you can kind of guess because you know that other plasmids have these specific sequences, and then you go from there by counting, right? If you know that other plasmids have the UR3, the ARS, this, that, then you can say it's about 6 kb, but then apparently there's more stuff in this guy here. So it says 10 to 15. So yeah, okay. Not bad, better than ChatGPT. So the problems with the YAC, the problems with the yeast artificial chromosome is that there are issues with stability. As I said, human genome has a lot of duplicate DNA, triplicate. It has a lot of repeats, the microsatellites that I talked about, the mini satellites, etc. And so what you're going to have is that if you use the yeast artificial chromosome and put it into yeast, you have more chances of creating homologous recombinant DNA molecules, and you don't want that to happen. Because if you have your chromosome inside the yeast, and then yeast is very good at homologous recombination, your chromosomes can be shuffled inside the yeast. So this is not the best way to do it, so the repeats can be unstable. This works for fragments that are very unique in sequence, but not fragments that have, let's say, 24 microsatellites, one after the other, okay? If the insert is large, then the yeast origin may not be sufficient to copy it. So yeast origin is fit for a specific amount of DNA. Yeast chromosomes are usually in the million, less than a million base pairs. So if you're adding already 500 kb, then you're starting to get close to that limit. And so it's not as efficient as origins of replication for humans, let's say. Obviously, if you have two inserts in the same cell, they might recombine if they have the same microsatellites, for example, and that will lead to a lot of repetitive sequence and then fragments that are shuffled between different sections. The solution is actually these bacterial artificial chromosomes, also known as BACs. And they're based on the F plasmids, so just like phosmids. They can clone up to 350 kb kilobases. So this is the one that's used for the Human Genome Project. They're a bit more stable than the yeast artificial chromosome because, you know, you can have recombination defective E. coli, and so that prevents that issue with the stability of your inserts, because if you as I mentioned, yeast is very good at recombination, and so because of the human genome being highly repetitive in nature, then that is a problem, because then there's a shuffling of our inserts that happens due to recombination. But you can have a recombination defective E. coli to prevent for the shuffling of the insert in the F plasmid. So the F plus plasmid is able to replicate the whole genome from bacteria. If you remember conjugation and horizontal gene transfer, where the F plus strain will transfer its DNA to the F minus strain. So in this case, you can technically transfer the whole genome of the E. coli, which is about 4.6 megabases, so 4.6 million bases. In general, though, that does not really happen. What happens is that you transfer about 350 kb, and then your transfer stops there. Okay, so this is the optimal size of your fragment, and the process is pretty much the same. You'll cut your F plus plasmid. You can cut it within the LAC-Z gene or whatever, so that you can select for it later, and then you'll insert. Your insert is cut with the restriction enzyme, and then you just ligate both together, put it in bacteria, and the bacteria will be plated. And then obviously you can use a marker as LAC-Z, where if you add XGAL, only the colonies that have an insert will appear white, because you would have disrupted the LAC-Z gene. Those that are blue mean that they have a wild type LAC-Z gene in the back, so that means there was no insert in the middle of the gene. And then you can select obviously your clones based on that. Because I'm re-recording this section here, I think we already answered this question. I'll just say the answer right here. So basically the backs don't have centromeres and telomeres because a bacterial artificial chromosome is of bacterial origin, and the composition of the nucleus of the yeast is not the same as the prokaryotic cell, which does not have a nucleus, by the way. And their transcription, translation, and replication are totally different. So bacteria usually have one circular chromosome. They don't have multiple chromosomes that are linear in nature. So the back does not have centromeres or telomeres, because these are structures of more complex organisms, such as eukaryotes. So we were talking about optimizing certain things, including the vector that you'll use to carry your fragment, but you also have to optimize your way of fragmenting DNA. And we already talked about this multiple times, so I will not cover it into too much. But basically, you need to find fragments that are of the right size so that you can then insert them in the right vector. And there are two ways to do that: either by partial restriction digest, where you're cutting with restriction enzymes, but not too much to have very small fragments, and not too little to have too big of fragments. You'd have to optimize the right way. The only one that I want to focus on is the physical shearing of the DNA. So we just saw here restriction enzyme shearing. Here it's physical. Because of the nature of the physical shearing, you'll produce these blunt-ended DNA molecules, so they don't have sticky ends. And so when you're breaking them, you need to add these linkers at the end, which will act as your sticky ends, right? And then these linkers usually are the opposite sequence of your sticky ends for the plasmid. And so you will need to, so if, let's say, for example, you break your DNA using sonication, vortex, or you push the DNA through a needle, which breaks it, then to allow it to ligate with the plasmid or the vector of interest, you will repair the ends by adding a linker here, which will make it look like this, and this is more fit to be inserted into your vector of interest. This part here just says that this technique gives you the most random libraries. It's somewhat true. So in this case here, the library is not random, it's pseudo-random, because the cut sites are known within are the same, they don't change within the genome, and so it's kind of random, because sometimes a cut site could be targeted or not, depending on how long you let the reaction happening. But if you do it a million times, it will be the same thing. While this one here, what happens is that the cuts are mostly random, because what happens is that you're using physical breaking of the DNA, and there's no sequence that is predictable, really. Although it's not 100% true, because what happens is that some sections of the DNA will be harder to break than others. For example, if you have GC-rich regions which are more solid than the AT regions, then what happens is that those will be cut less. But the impact of this on the randomness is less than that of the restriction sites. And so, a few considerations to build the library that you have to think about before actually jumping in. So we optimized the shearing, we optimized the vector, but you need to actually think about your library before jumping into it. Your library has to reflect the genome from which it was made. And so if you're looking at a strain in the laboratory, you'll take the most common strain because that's the one that is used the most, and you'll use that as a source of your genomic DNA to make the library. But for humans, it's important to actually take DNA from multiple groups around the world and not just from a certain group, because what happens is that even though we have similar DNA, similar genomes, we have changes in our DNA that are different based on the location where we are in the world. And these could be just SNPs, they could be deletions, they could be translocations, things like this. So we have to be comprehensive in combining the DNA from multiple people to get a good representation of what is an actual human genome. And for example, for the Human Genome Project, they used a mix of DNA from individuals of different ethnic backgrounds. And then they sequenced all that, and using computers, they compared the sequences to kind of figure out what is the actual human genome, irrelevant of the ethnic background. For the Celera Genomics project, they started with the DNA of Craig Venter himself, but then they joined with the Human Genome Project, and then when these guys sequenced DNA, Celera was using it also in its studies. Here I'm representing the normal genome, so I'm not saying that different ethnic backgrounds don't have a normal genome. What I mean is more like the sequence of the genome that is not really specific to one group or the other. Okay. Other considerations if you're studying, for example, when you're screening the X chromosome, you have to consider the role of sex-linked chromosomes as a DNA source. So if you're screening a male, then you have to screen twice as many clones because males don't have 2 copies of the X chromosome. Females do have 2 copies of the X chromosome, and because of that, you need to account for that. Just like for the autosomes, which are duplicate, etc., you have to think about that. And obviously, you have to make sure that your cells are quite young, because as time goes through, you already know, I'm sure you guys know this, but you accumulate mutations in your DNA, deletions, translocations. So you want to avoid using a lineage that is not representative of the actual normal genome, let's say. A cell lineage that is that was not affected by, let's say, mutations through time, right? And so you need to take some sort of measures to get your, let's say, young samples, which are less mutated and less modified through different forces through time. Although this point here is, it's important to think about it, because then if you take only young cells or younger cells, then what happens is that you'll have a genome representing the younger population, for example, but not necessarily the older population. And then you start doing research on, for example, aging, which is more a older people kind of, the genome of older people is more representative of it, or at least the result of aging is more represented in the older population. And so this is a little bit tricky here, but this is how it was done. This part here I presented in the next lecture, so we'll just skip it. This 2, this 2, this 2, this 2, So the homework was given to you guys already, and you all submitted this. No, you will be submitting this homework this week. Finally, for the team presentations, I'll just repeat this because this is important. So each person needs to find 2 people to work with, so that's a team of 3. Send me an email with the name of the people that are in the team, then send me a second follow-up email where you'll tell me which paper you'll be working on. I need to approve the paper to make sure that the paper is of the right level. The paper can cover pretty much anything from genomics of protists to I don't know what. Okay, as long as it's about genomics, try to find a paper that's not too old, because if you go to the older papers, they have like 1 experiment in it and that's it. I want something recent so that we can have a good level of science in it. Try to understand the paper so that you're able to kind of present the science behind it accurately. So I just need a presentation about the claims of the paper, some background overview for the class because they don't know, for example, that topic, so that they have an idea of the theory they need before getting into the results. Explain the methods, explain the limitations of the paper, things like this. You have to prepare a 25 to 30 minute presentation, and so that gives you about 10 minutes of presentation per person. One thing that I mentioned that I'm re-recording right now is the fact that I have been a student recently, so it's not a long time ago, and I know how teamwork works. So you guys are pretty advanced, so that should not be a problem, but I still have to do this disclaimer. If there is one person that is not working in the team and the other 2 complain about that person, and I make my verifications and find that person not working, I will make sure that that person presents all the paper by herself or himself. The reason for that is that I don't want to jeopardize people in a team that are trying to actually succeed because of another person. So I really hate that. Actually, just a little story. I used to be the person doing all the work in a team, so now it's time to correct this mistake. If one person of the team misses the presentation, the others will still present. You need to be able to present everyone's part in this presentation. What else? So make sure that you are able to do the presentation by yourself, no matter what the situation, okay? And obviously, if you miss the presentation, medical note and all that stuff, I think you guys already know this. Try to not though, because you don't want to add up more work just like that. So just do your 10 minutes and that's it. The audience will be evaluating the papers based on a rubric that I'll provide. This is not included in the grade, so I didn't like professors doing this in the past, and I'm not gonna do it. But I hate the fact that some professors make you grade other students as a student and then include their grades in your grade. I'm not doing that. This is just for me to first of all, as a participation exercise, second, so that you can get some practice in terms of evaluating science and presentations. One thing though that is important is that you will be providing a multiple choice question that tests the paper or the presentation at least, so that science is not gray, black or white. It's more like gray in the sense that we do experiments and things like this, but there's always place for optimization. There's always things that we didn't think about. So I would like each time the whole class, so each student will provide me for a multiple choice question about a paper that is being presented. So if there is 6 papers being presented, that's 6 multiple choice questions. Make sure to be there because these count for your participation. And will also be a great exercise to kind of see how others are doing their work, and at the same time how you could improve your own presentation based on that. With that being said, I'll see you next class. Take care.